Hello and welcome to the Microorganism Cultures revision video from scienceworksheets.co.uk. Please like, subscribe and comment to support the channel. Bacteria are a type of microorganism. Bacteria divide by a process called binary fission. Conditions for culturing microorganisms. Bacteria are grown in a nutrient broth solution or as colonies on an agar gel plate in a lab. They need nutrients such as protein and carbohydrates. Warmer temperatures will increase the population growth, but temperatures above 25 degrees C are more likely to encourage the replication of pathogenic bacteria, which can cause us harm. Under ideal conditions of warm temperature and a good supply of nutrients, the bacterial population can double once every 20 minutes. Preparation of an uncontaminated culture using aseptic techniques. Aseptic conditions mean that the conditions are sterile and there is no contamination present from other microorganisms. If other microorganisms are present, they could compete for nutrients with the ones that you are trying to culture. They could also be pathogenic and cause us harm. And the following steps are taken to ensure aseptic conditions. A Bunsen burner with a blue flame is used to create a sterile zone, the area around the Bunsen burner flame, where the rising convection currents help keep airborne microbes and dust away from your work. The agar or broth medium, along with the petri dishes, all need to be sterilized prior to use. Sterilization of the agar or broth is done using an autoclave. This uses high pressure steam at 121 degrees Celsius to kill any bacteria. Glass petri dishes can also be autoclaved for sterilization. Plastic petri dishes are sterilized using UV light. Agar or nutrient broth cannot be sterilized using UV light, as it will not penetrate them. A metal inoculating loop is sterilized by holding in a blue Bunsen flame until it is red hot. It is then allowed to cool in air before the next stage. The cool, sterilized loop is dipped into a suspension of microbes and then streaked across the surface of the agar in a petri dish. The lid of the dish should be left ajar to try to keep the area sterile while streaking. Remember to keep working within the sterile zone of the Bunsen flame. After streaking, the lid of the petri dish is fixed in place with 2 pieces of tape. It is important not to completely seal all around the dish. This is to allow oxygen to enter the petri dish. If oxygen cannot enter, it will increase the chance of anaerobic bacteria growing, which are more likely to be pathogenic. The dish should then be incubated at 25 degrees C upside down. Upside down prevents condensation falling on the agar. Higher temperatures are used in industry or hospitals. In schools and colleges, the maximum temperature is 25 degrees C to reduce the risk of pathogenic bacteria growing. Thank you for watching. See our revision notes on this topic. The link is in the description.